Chapter 1, Cellular and Molecular Biology of Aqueous Hemodynamics, from Shields' textbook of Glaucoma, 6th edition, page number 8. Cellular organization of the ciliary body and the ciliary processes. The ciliary body is one of three portions of the uveal tract or vascular layer of the eye. The other two structures in the system are the iris and choroid. The ciliary body is composed of A. Muscle, B. Vessels, C. Epithelia lining the ciliary processes, and D. Nerve terminals from the autonomic nervous system. Ciliary body muscle. The ciliary muscle consists of two main portions, the longitudinal and circular fibers. The longitudinal fibers attach the ciliary body to the limbus at the scleral spur. This portion of muscle then runs posteriorly to insert into the suprachoroidal lamina as far back as the equator or beyond. The circular fibers occupy the anterior and inner portions of the ciliary body and run parallel to the limbus. One-third portion of the ciliary muscle has been described as radial fibers, which connect the longitudinal and circular fibers. The physiologic function and pharmacologic action of parasympathomimetic agents as they relate to the ciliary muscle are discussed in Chapter 32. So, what we learned in this paragraph is the ciliary body muscles are of three types: longitudinal, which are also known as meridional, and they extend from scleral spur. They get inserted into the scleral spur and are posteriorly attached to the suprachoroidal lamina. Then, second fiber type is radial, or also known as oblique fibers. They connect this phrase to, that is longitudinal or meridional, to the circular fibers and circular fibers. So ciliary body muscles are of three types: longitudinal or meridional, second radial or oblique, and third circular. Ciliary body vessels. On the basis of studies in primate and human eyes, the vessels of the ciliary body appear to have a complex arrangement with collateral circulation on at least three levels. The anterior ciliary arteries on the surface of the sclera send out lateral branches that supply the episcleral plexus and anastomos with branches from adjacent anterior ciliary arteries to form an episcleral circle. B. The anterior ciliary arteries then perforate the limbal sclera. In the ciliary muscle, branches of these arteries anastomose with each other as well as with branches from the long posterior ciliary arteries to form the intramuscular circle. Divisions of the anterior ciliary arteries also provide capillaries to the ciliary muscle and iris and send recurrent ciliary arteries to the anterior choriocapillaries. C. The major arterial circle lies near the iris root, that is anterior to the intramuscular circle, and is actually the least consistent of the three collateral systems. Although the primate studies reveal a contribution from perforating anterior ciliary arteries, non-primate animals indicate that this circle is formed primarily, if not exclusively, by paralimbal branches of the long posterior ciliary arteries, which begin dividing in the anterior choroid. In any case, the major arterial circle is the immediate vascular supply of the iris and ciliary processes. Each ciliary process in primates is supplied by two branches from the major arterial circle, the anterior and posterior ciliary process arterioles. Anterior ciliary process arterioles supply the anterior and marginal, that is innermost aspects of the major ciliary processes. These arterioles have luminal constrictions before producing irregularly dilated capillaries within the processes, suggesting precapillary arteriolar sphincters. This may represent the anatomic site of adrenergic neural influence on aqueous humor production by regulation of blood flow to the ciliary processes. The posterior ciliary process arterioles supply the central, basal, and posterior aspects of the major ciliary processes, as well as all portions of the minor processes. These arterioles are of larger caliber than the anterior arterioles and lack the constrictions seen in the lateral vessels. Both populations of arterioles have interprocess anastomosis. Vascular casting studies of capillary networks in the ciliary processes of human eyes suggest three different vascular territories with discrete arterioles and venules. The first is located at the anterior end of major ciliary processes and is drained posteriorly by venules without significant connections to other venules in the ciliary processes. The second is in the center of major processes, whereas the third capillary network occupies the minor processes and posterior third of the major processes. Both of the latter territories are drained by marginal venules which are situated at the inner edge of the major processes. It is thought that these three vascular territories may reflect a functional differentiation in the process of aqueous humor production. Venous drainage is into choroidal veins either from the posterior aspects of the major and minor processes or by direct communication from the interprocess connections. So, from ciliary body vessels, from these paragraphs, what we learned the ciliary blood vessels, the arterial supply. So, it is from anterior ciliary arteries and long posterior ciliary arteries. These two form the major arterial circle that lies near the root of the iris and that is anterior to the intramuscular circle. Now what is this intramuscular circle? It is intramuscular means it is inside the ciliary muscles. So this intramuscular circle is nothing but the anastomosis of anterior ciliary artery branches with each other and with branches from the long posterior ciliary arteries LPCM. So this anastomosis circle in the ciliary muscles, it is called as intramuscular circle. So this major arterial circle, it lies near the root of the iris anterior to this intramuscular circle. So major arterial circle, it is located near the root of the iris anterior to the intramuscular circle, and then now there is ciliary process. Okay, so each ciliary process is supplied by anterior ciliary process arteriole and posterior ciliary process arterioles. These anterior and posterior ciliary process arterioles are given off from the major arterial circle. So major arterial circle, it gives rise to two branches, anterior ciliary process arterioles and posterior ciliary process arteriole. So these two supply each ciliary process, and this anterior ciliary process arteriole, it supplies anterior and marginal, that is innermost aspect of the ciliary process, major ciliary process, and they have precapillary arteriolar dilatations or sphincters, and that is the anatomic site for adrenergic neural influence, and that affects the or regulates the aqueous humor production by regulation of blood flow to the ciliary processes. Now this posterior ciliary process arteriole, that is from MCA, that is major arterial circle, It supplies the central, basal and posterior aspects of major process, and it also supplies all portions of minor ciliary processes. And these posterior ciliary process arterioles, they have got a larger caliber than anterior ciliary process arteriole, and they lack dilated precapillary sphincters. These both arterioles, they have interprocess anastomosis. Now next is ciliary processes. The functional unit responsible for aqueous humor secretion is the ciliary process, which is composed of capillaries, stroma, epithelium. The ciliary process capillaries. The ciliary process capillaries occupy the center of each process. The thin endothelium has false porous area of fused plasma membranes with absent cytoplasm, which may be the site of increased permeability. A basement membrane surrounds the endothelium, and neural cells or pericytes are located within the basement membrane. A very thin stroma that surrounds the capillary networks and separates them from the epithelial layers. The stroma is composed of ground substance consisting of mucopolysaccharides, proteins, and plasma solutes, except those of large molecular size. Very few collagen connective tissue fibrils, especially collagen type 3, and migrating cells of connective tissue of blood origin. And migrating cells which are of connective tissue and blood origin. Tubular microfibrils with and without elastin have been demonstrated in bovine ciliary body, especially in the stroma of the pars plana in relation to zonules. Two layers of ciliary epithelium surround the stroma, with the apical surfaces of the two cell layers in opposition to each other. The pigmented epithelium has numerous melanin granules in the cytoplasm and an atypical basement membrane on the stromal side. In the non-pigmented epithelium, the basement membrane is composed of glycoproteins that are immunoreactive for laminin and collagen types 1, 3, and 4. This membrane, which faces the aqueous humor, is also called the internal limiting membrane and fuses with the zonules. The non-pigmented epithelium stains less intensely than the pigment layer for cytokeratin 18, but more so for vimentin, with a predominant distribution in the crests of the pars placata and the posterior pars plana. It also stains with antibodies against S100 protein, another molecule with restricted expression in the non-pigmented cells are the water channels aquaporin 1, which is also expressed in trabecular endothelium, and aquaporin 4. In transgenic knockout mice which do not express these water channels, IOP is significantly reduced compared within the wild-type mice whose water channels are normally expressed. The mechanism of IOP lowering is due reduction in decreasing aqueous humor production, but not in outflow. Although these genetically modified mice have phenotype of lower IOP, patients with aquaporin 1 mutations have normal IOP. A variety of intercellular junctions connect adjacent cells within each epithelial layer as well as the apical surfaces of the two layers. Such junctions include gap junctions, which are expressed by the pigmented cells, the non-pigmented cells, and the pigmented non-pigmented cells, and tight junctions or zonular occludens, which are expressed between the non-pigmented cells. It is primarily the zonular occludens in the non-pigmented ciliary epithelium that creates an effective barrier to intermediate and high molecular weight substances such as proteins. Electrophysiologic studies of rabbit ciliary epithelium suggest that all of the cells in the epithelium function as syncytium. Tight junctions create a permeability barrier between non-pigmented epithelial cells which forms part of blood aqueous barrier. These tight junctions are said to be the leaky type in contrast to the non-leaky type in blood retinal barrier and may be the main diffusional pathways for water and iron flow. Microvilli separate the two layers of epithelial cells. In addition, ciliary channels have been described as spaces between the two epithelial layers. These channels may be related to the formation of aqueous humor in that they develop between the 4th and 6th months of gestation, corresponding to the start of aqueous humor production. So, this description about ciliary processes can be summarized in this diagram. So in this diagram you can see at the center there is a blood capillary. It is lined by the endothelial cells, and this capillary endothelium has vast porous area of fused plasma membrane with absent cytoplasm, and it may be the site of permeability. So after that, this is the basement membrane of the capillary blood. Then after that, in between the pigmented epithelial and non-pigmented epithelial layers and the blood capillary, there is a thin stroma, and this thin stroma is made up of mucopolysaccharides. The ground substance of the thin stroma is made up of mucopolysaccharides, proteins, plasma solutes, except for those large molecular size proteins. Then it's made up of the collagen fibrils, mainly type 3, and migrating cells which are of connective tissue and blood origin. Then after that comes the basement membrane of the pigmented epithelial cells. Then comes pigmented epithelium. Now this pigmented epithelium, it has got melanin granules, and it has got gap junctions. It has got sonula adherens, desmosomes and gap junctions. Then after that comes the non-pigmented epithelial layer. Now this non-pigmented epithelial layer, it has got well-developed tight junctions, sonula occludens. They create an effective barrier that is called as blood aqueous barrier, but this blood aqueous barrier is leaky. That is, it allows the passage of water and ions to them, but blood retinal barrier is non-leaky. That's the difference. Now, in between these pigmented and non-pigmented epithelial cell layers, there are microvilli. Okay, so the basement membrane of non-pigmented epithelium, it faces towards the aqueous humor, that is the posterior chamber. And this basement membrane is also called as internal limiting membrane, and it fuses with the sonules of the lens. And this non-pigmented epithelium, it stains less intensely with cytokeratin 18 and more with vimentin as compared to the pigmented epithelial cells. Now this ciliary process is the functional unit that is responsible for the aqueous humor secretion. It's a functional unit of aqueous humor secretion. And yeah, so this non-pigmented epithelium also has aquaporin 1 channels, but their function is not yet clear because patients with mutations in aquaporin 1, they have got normal IOP. And the space between the pigmented and non-pigmented epithelial layers is called as ciliary channel. These are called as ciliary channels, and spaces between the non-pigmented and pigmented epithelial layer are called as ciliary channels, and they may be related to formation of aqueous humor because they develop between 4 to 6 months of gestational age that corresponds to aqueous humor production. So that is the summarization of the ciliary process. Now the autonomic innervation of the ciliary body. Both sympathetic and parasympathetic nerve endings innervate the ciliary body. The parasympathetic fibers synapse in the superior cervical ganglia, and the postsynaptic fibers are distributed to the ciliary body vessels. Because the ciliary epithelium is not innervated, It is thought that the catecholamine neurotransmitters released from the sympathetic nerve endings diffuse to the adrenergic receptors on the ciliary epithelium. Stimulation of these receptors increases aqueous humor secretion by the ciliary epithelium, which is discussed further in the section on molecular mechanisms and regulation of the aqueous humor production. The parasympathetic fibers originate from the Edinger-Westphal nucleus, that is of the oculomotor nerve, third motor nerve, cranial nerve. And it is to innervate the ciliary muscles. Stimulation of these nerve fibers releases acetylcholine, which then stimulates the cholinergic receptors on the ciliary muscle. These activated receptors cause the ciliary muscle to contract, causing accumulation by changing the shape of the crystalline lens. In addition, ciliary muscle contraction reduces resistance to conventional aqueous humor outflow or trabecular outflow and may also affect unconventional aqueous humor outflow or uveoscleral outflow. The effect of the cholinergic pathway on the trabecular outflow pathway is used pharmacologically in the treatment of glaucoma, and it is discussed in chapter 32. So these two paragraphs can be summarized as yeah. Okay. The autonomic innervation of the ciliary body is sympathetic and parasympathetic. The sympathetic, the synapse in the superior cervical ganglion, and they travel along the ciliary blood vessels and reach to the ciliary body vessels. And these vessels, they have got precapillary sphincter dilated arterioles at the commencement of capillaries in the ciliary processes, and that may affect the aqueous production by affecting the blood flow to the ciliary processes. Then the ciliary epithelium is not innervated. So it is thought that catecholamine neurotransmitters diffuse to these epithelial cells. When stimulated, they are responsible for aqueous humor production, increase in the aqueous humor production. The parasympathetic nervous system to the ciliary body, it originates from Edinger-Westphal nucleus. It is of third cranial nerve, that is oculomotor nucleus, and stimulation of which it releases catecholamines. Stimulation of which releases acetylcholine, and this acetylcholine stimulates ciliary muscles. They contract, and it is a part of accumulation, and it is responsible for increasing uveoscleral outflow by pulling the scleral skull posteriorly, opening up the angle, opening up the trabecular major places, and also it is thought to be increasing the uveoscleral outflow, unconventional outflow. Figure 1.4, schematic of the three major components of the ciliary body. 1. Ciliary muscle, composed of longitudinal LCM, radial and circular CCM fibers. The vascular system formed by branches of the anterior ciliary arteries ACA and long posterior ciliary arteries LPCA, which form the major arterial circle MAC. So major arterial circle is formed by anterior ciliary arteries and long posterior ciliary arteries, branches from these two. And 3. The ciliary epithelium CE, composed of an outer pigmented and an inner non-pigmented layer. Composed of an outer pigmented and an inner non-pigmented layer. So the ciliary body composition is given in this figure 1.4. It's made up of muscles, vessels and epithelium. Epithelium is pigmented, non-pigmented. Muscles are of three types, that is longitudinal, meridional Second is oblique or radial. Third is circular. And the vascular system, that is coming from the anterior arteries and long posterior artery branches, forming the major arterial circle in the root of the iris anterior to the intramuscular circle. Now figure 1.5, schematic of vascular interconnections of two contiguous major ciliary processes, lateral anterior arterial branches join to form interprocess capillary networks arrowhead, which provide communication between major processes, laterally directed posterior arterioles from posterior interprocess networks through which the minor ciliary process receive blood. In addition, both anterior and posterior interprocess networks drain directly into the choroidal veins. So in this diagram, we can see the anterior arteriole. So this is the major arterial circle, okay, MAC. It gives off anterior ciliary process arteriole that supplies the anterior aspect of the major ciliary process, and then there is this posterior ciliary process arteriole, which supplies the posterior aspect, basal aspect, and then minor processes, central aspect and minor ciliary processes. And in between these two there is interprocess anastomosis, and that drains into the choroidal veins directly. And then we can see here the minor ciliary process receiving the blood supply from the posterior ciliary process arteriole. We can also see the choroidal veins here posteriorly. So this is the anterior side, this is the posterior side. Sorry. Figure 1.6, light microscopic view of ciliary processes, section perpendicular to radial ridges, showing major ciliary processes and minor ciliary processes from a human eye stained with toluidine tube. Now figure 1.7, schematic of the ciliary epithelium summarizing the histology and junctional complexes. A. Physiology of ionic transport mechanisms. B. Transmembrane signaling and enzymatic pathways and other paracrine functions. C. Now description of figure A. The ciliary epithelium is composed of two layers containing nuclei and with an outer pigmented layer facing the stroma of the ciliary process and inner non-pigmented layer facing and lining the posterior chamber. Apical surfaces are in opposition to each other. Basement membrane BM lines the bilayer and constitutes the internal limiting membrane on the inner surface. The non-pigmented epithelium is characterized by mitochondria, zonula occludens, and lateral and surface interdigitations. The pigmented epithelium contains numerous melanin granules. Additionally, intercellular junctions include desmosomes T and gap junctions G. Now the description of figure B. Overall, there is a net secretion of cations, open arrows. These cations are sodium, potassium and hydrogen ions, and anions, chloride and bicarbonate anions, HCO3-. But there is also some absorption, solid arrows of these ions. The net effect is a negative charge toward the posterior chamber relative to the ciliary body stroma. The transfer of these ions proceeds primarily through a transcellular route or transport across the bilayer through some ion channels and transporters, black rectangles. Transfer also occurs to a lesser extent through the paracellular route or between the cells. Now description of figure C. Aqueous humor secretion is highly regulated by multiple transmembrane receptor mediated pathways. GPCR stands for G protein coupled receptors, G for G protein, AC for adenylate cyclase, ATP stands for adenosine triphosphate, CAMP means cyclic adenosine monophosphate, PLC for phospholipids, C. PR is for phosphatidylinositol, DAG means diacylglycerol, IP3 stands for inositol triphosphate. Enzymatic mediated pathways, CA for carbonic anhydrase type 2 and possibly type 4, and specialized transporters such as the aquaporin type 1 channel AQP1, which has restricted expression in the non-pigmented ciliary epithelium. The precise localization to pigmented versus non-pigmented and orientation on apical versus basolateral surfaces are unknown for these pathways. Thus, they are represented in a biennial couplet. Other potential paragram functions of the ciliary epithelium include secretion of small peptides, granules. Thank you. Gosh.